This is a production of Cornell University.
Thank you so much, Mike, for that resounding vote of confidence. So today I'm going to talk to you about kind of my RNA-centric view of how plants respond to stress or changes in their environment. And so I guess when most of us think about RNA in terms of stress responses, we really think of their steady state levels. We're imagining that these RNAs, or the abundance of these RNAs, are really a proxy for the protein. So of course, you have changes in the environment eliciting some gene to produce an RNA that is then producing a protein, and then that is the response that we're really thinking about. And so RNA is really just this intermediate, this messenger. While a lot of very interesting biology and molecular mechanisms have come about from this perspective, I would argue that there are a number of ways in which RNAs act that don't depend on producing a protein. So there are the long non-coding RNAs and there are the small non-coding RNAs. And while many of them do kind of interfere or regulate the production of a protein, many of them can elicit a response in their own rights without ever producing or regulating protein production or translation. And so some of the RNA-mediated responses that I'm interested in, I have listed here, and I'm going to talk about several of them today. But the first one is going to focus on, as Mike mentioned, the long non-coding RNA class of RNAs, for which we know relatively little in all of eukaryotes, really. Okay, what are long non-coding RNAs? Long non-coding RNAs are these transcripts that are greater than 200 nucleotides, for which we have little evidence of protein synthesis, so no strong open reading frame. These transcripts can arise really from anywhere in the genome, which means they can be transcribed from intergenic space between two other genes, but they can also be transcribed in the antisense direction overlapping a known gene, or actually even intronically within a known protein coding gene, for instance. And most of these transcripts, most of these lincRNAs are thought to be Pol2 transcribed. So they're going to have a 5' cap and a poly A tail. And this is important when it comes to identifying them using RNA sequencing data, because since they look just like mRNAs and we're capturing mRNAs, we're going after mRNAs primarily in our RNA sequencing data. That means we can pull out or identify lincRNAs from these data as well. LincRNAs typically display poor sequence conservation, and this really, I think, is one of the main hindrances in terms of identifying lincRNAs in close relatives of model systems in which we've identified lincRNAs to begin with. So poor sequence conservation, and I'll talk about this a little bit more later as well. In terms of function, we know that lincRNAs, or we often think of lincRNAs primarily in their role as a transcriptional regulator. So lincRNAs are often regulating the transcription of genes either in cis or in trans. We know that lincRNAs can also regulate production of or protein synthesis through modulating how microRNAs are binding to mRNAs. LincRNAs, through sequence complementarity, can actually recruit away microRNAs from their intended target and allow for protein synthesis to occur. LincRNAs also are commonly known as modifiers of the epigenetic state, particularly of the chromatin around the locus that they're being transcribed from. And this usually occurs through the recruitment of the polycomb repressor complex, PRC2, which then comes in and lays down chromatin marks. And lastly, lincRNAs are known to act as these scaffolds, these RNA scaffolds that recruit proteins that wouldn't normally interact together into a single complex that then has either enzymatic or some kind of molecular function. And one important example of this is the telomerase RNA, which is an RNA I studied as a graduate student in Dorothy's lab. And this RNA brings together a whole suite of proteins that don't physically interact directly, but together or through this scaffold RNA. They come together and they maintain chromosome ends in eukaryotes. I should mention here that only a few lincRNAs have been functionally characterized in plants, particularly in Arabidopsis, where most of our knowledge about long-encoding RNAs comes from. And this is in stark contrast to the hundreds of lincRNAs that have been functionally annotated in mammals. So here I'm just listing a few of the annotated lincRNAs. Again, most of these are coming from Arabidopsis, but it's a very small number. And I'd like to unpack why that is, or why I think that is. And to describe that, or just describe this disparity, I'm showing you a phylogeny representing most angiosperms. So eudicots here, a couple of asterids, and some monocots, along with what we call these more basal species, humans and mice. And if we just look at the number of lincRNAs that have been published for each of these species, we see that there are over 100,000 lincRNAs that have been identified, annotated in humans and mice each, compared to roughly about 6,000 or so lincRNAs that have been published in Arabidopsis. And I would argue that this disparity arises from the millions of dollars that went into the human and mouse UNCODE projects that really performed an enviable and exhaustive annotation of all the functional elements of these two genomes. Where lincRNAs have been identified in plants, different criteria and different experimental evidence were used in their identification. For instance, in Arabidopsis thaliana, most of the lincRNAs that we have annotated in that genome were actually identified using tiling array experiments. And in contrast, the lincRNAs identified in some of these other species, like corn, for instance, were identified using RNA sequencing data. And on top of the different kind of technologies that were used to identify these lincRNAs, different criteria or filtering metrics were used to determine or to infer whether or not the transcript was a lincRNA. So there's really kind of been this grab bag approach, hodgepodge approach to identifying lincRNAs across the plant lineage. What we want to do is utilize, what we are in the process of doing, I should say, is to utilize the available RNA sequencing data that's available across the plant lineage that really, I would argue, brings some level of parity between plants and mammals, where we have a huge amount of RNA sequencing data present for Arabidopsis and corn, and I would say, a decent amount in each of these other species that allows for an exhaustive curation efforts without I would say, millions of dollars that went into the human and mouse UNCODE projects. So we want to identify lincRNAs from these RNA sequencing data. As I mentioned, we can identify lincRNAs, most of them, because they look just like mRNAs in terms of RNA-seq library preparation. So these RNA sequencing data are a perfect resource. And we are going through and we're in the process of comprehensively analyzing and identifying lincRNAs from these RNA sequencing data. And today I'm going to talk a little bit about our work, just a little snapshot of some of our work in the Brassicaceae lineage. Now, just to orient you for those of you who aren't familiar with Brassicaceae, obviously we have the model system Arabidopsis thaliana. Here in this system, in this family, we decided to go after long-encoding RNAs in 3 other species to help us develop a better comparative system. One of those species is Camelina sativa, which is known as a plant with high-quality oil in its seeds, as well as Brassicaceae, which we're all quite familiar with, and Eutrima salsigenea, which is a salt-tolerant species within the family. There's a decent amount of RNA sequencing data for each of these species. Obviously, Arabidopsis thaliana has the most, 16,000 unique SRAs. We processed all of these RNA sequencing data through a couple of computational workflows that we developed. One is called RMTA, which is used to actually process all of these RNA sequencing data in high throughput in a distributed workflow or a distributed manner, so that we can essentially just start a job and walk away, and all of our RNA sequencing data is processed. And then we took all the processed RNA sequencing data, all the transcripts that we identified, ran them through our lincRNA identification pipeline EvoLink, which not only identifies link RNAs, but also assesses the degree to which link RNAs are conserved across a particular lineage. And then we identified link RNAs. We initially identified about 21,000 long encoding RNAs in Arabidopsis thaliana. These are supplementing the previous data sets or the previous annotation for Arabidopsis. And then we also identify a similar or greater number in each of the other species. Now, one of the things that we did on top of just merely identifying these link RNAs and saying that each of these transcripts appear to be non-coding is we applied some additional hierarchical filters that have been applied in mammalian systems to get at a short list of more high confidence long encoding RNAs. And we can talk about what each of these filters is afterwards if anybody's interested in getting into the weeds. But in the end, we get this list of about 10,000 Arabidopsis long encoding RNAs. And I should emphasize here that about 1,900 of these link RNAs overlap or coincide with previously identified link RNAs identified in Arabidopsis using titling array technologies. Okay, so now that we have link RNAs, we've identified them, what do we do now? Because we're not just doing this for the sake of identifying novel transcripts. We'd really like to get down to some kind of function. And to identify or to infer link RNA function, we're taking advantage of the comparative framework that we've set up in Brassicaceae and that we're applying to each of the major plant lineages that in which we're identifying link RNAs. And we're using these comparative frameworks to address two questions. The first of which is, should I care? Is this transcript functional? And the second is, where do I start looking for a function? And to get at if we believe a transcript is functional, we're using three different metrics. We're looking at sequence conservation, structural conservation, and tissue level transcriptome conservation. And because we have 4 species in which we've identified link RNAs and we can compare between each of these species, when we do pairwise comparisons, looking at each of these 3 metrics, we get essentially a set of link RNAs for which we have high confidence that they're sequence conserved, they're structurally conserved, and they appear to be conserved at the transcriptomic level. And then we take that list of link RNAs and we ask our second question, which is where do we start looking for function? We have this short list of link RNAs, what do we do now? And we get at, we infer function based on associative techniques. For instance, we're using transcriptomic guilt by association, like weighted gene co-expression networks. We're looking to see if any of our link RNAs share domains with known functionally described link RNAs. It's a very short list right now, but we believe as we develop this, that will be a more promising way of getting a function. And then we're also just asking, are any of these link RNAs transcriptionally responsive to changes in the environment? So we have all of these stress experiments that are publicly available that the community has already performed. We're asking, are any of these link RNAs found to be responsive in these experiments? And then again, we perform the same kind of pairwise comparisons. Do we see a link RNA from species 1, say Arabidopsis, that is not only sequence conserved, but also appears to be co-expressed with the same types of genes in both species, the same stress responsive genes in both species? And in the end, what we get is this annotated table of all the link RNAs we've identified in each of these species and the degree to which they're conserved, the degree to which they're stress responsive, or the degree to which they're involved in different developmental processes based on these analyses. And we're using these, as I said, we're using these approaches now in the Brassicaceae, now that we have these link RNA data sets from multiple species. We can't do this if we couldn't do this if we just had link RNAs from Arabidopsis, for instance, because we would have nothing to compare against. Comparing between Arabidopsis and corn, for instance, is too far of an evolutionary distance. Link RNAs are just so poorly conserved that we can't make those types of analyses or comparisons. Okay, so first we just look at sequence conservation. And what I'm showing here is from a Arabidopsis thaliana perspective, the degree to which a link RNA can be traced back through evolutionary time, back to the base of Brassicaceae. And you'll notice straight off that roughly 43 or 44% of Arabidopsis lincRNAs are restricted to the Arabidopsis genus based on sequence conservation, in contrast to about 3% of protein coding genes that are restricted to the Arabidopsis genus. You can compare this to what we see when we compare between Arabidopsis thaliana and lineage 2, represented here by Brassicaceae, Shrinkiella, and Eutrema. We see about 27% of the Arabidopsis lincRNAs are conserved to this evolutionary time point. And then only 1%, roughly 1%, is conserved based on sequence similarity to Terranea hasleriana, which is a member of Cleomaceae, which is the sister family of Brassicaceae within the Brassicales. Again, contrasting this with the sequence conservation we see at the protein level, or at the within protein coding genes, roughly 43% of protein coding genes can be identified in between Arabidopsis and Terranea hasleriana. So take homes from this: about 30% of the Arabidopsis lincRNAs that we identified can be traced to node 4 here, to the coalescence point between Arabidopsis and Brassica or Eutrema, whereas 91% of Arabidopsis protein coding genes can be traced to that similar time point. We can also use these analyses to determine if our sets of lincRNAs are conserved as lincRNAs in other species. So here I'm showing, or I just want to focus on node 4 here. Again, the coalescence point between Arabidopsis thaliana and Brassicaceae or Eutrema. And about, I would say, more than half of the lincRNAs that are conserved at the sequence level between lineage 1 here and lineage 2 are conserved as a long encoding RNA. And then a large fraction are conserved at the sequence level to genomic elements for which we have no annotation at all. And then a very small subset are conserved as protein coding genes, or annotated as protein coding genes in those species, or in these two species. And so these, this subset of lincRNAs that are conserved as an mRNA and a close relative, these could either be evidence of pseudogenization, where we're now looking at a lincRNA that's undergone a pseudogenization event, it's no longer protein coding, or it could be misannotation of the lincRNA in Arabidopsis. And without going into too much detail, we're very happy that we see very few lincRNAs annotated as, or sharing sequence similarity with protein coding genes in other species. So to summarize, or I take home from this comparative approach, these roughly 4,000 lincRNAs that we've identified to be conserved between Brassicaceae, Eutrema, and Arabidopsis thaliana are promising candidates for functional analysis, promising candidates for which we can attempt to infer function based on these associative techniques I told you about. However, this approach, the sequence-based homology approach, is going to miss lincRNAs that are under different evolutionary constraints. And I want to unpack that a little bit more. I really think that this aspect is really interesting. So here I'm showing you kind of an alignment, a cartoon of an alignment between a region of a genome in species A, for which we've annotated a long encoding RNA, and a centinic region of a genome in species B. And by centinic, I mean the protein coding genes present in that in these two genomes are collinear. They share similarity, sequence similarity, and arrangement. And if we're just going based on a genomic perspective, in the absence of sequence conservation at this particular locus, we would say that there's no lincRNA in species B. However, we know, based on work in Arabidopsis from Martin Crespi's group and others, that some lincRNAs don't need sequence. They function in a sequence-independent manner. It's the act of transcription that's important. And now that we have lincRNAs that we've identified in each of these different species, we can go through and we can ask if a transcript, if a long encoding RNA transcript is arising from a locus, a centinic locus, for which we have very little sequence similarity. And I'm calling these types of lincRNAs transcriptional syntelogs, because they're arising from syntenic space, but they share very little sequence similarity. And again, this is only really possible because we have this comparative transcriptomic system where we have link RNAs identified in each of these species within a relative close evolutionary time frame. And just to, again, we're looking at the link RNAs for which we were able to identify transcriptional syntelogs. And just a take home here is that we were able to identify an additional 3,300 link RNAs that are not conserved at the sequence level between Arabidopsis IncRNA and Brassicaceae, but appear to be conserved at the transcriptional level. That is, they're conserved in the same genomic space. They share synteny, but they share very little sequence similarity. And just to give you an example of what I'm trying to talk about here, here's a Koji screenshot of chromosome 4 from Arabidopsis IncRNA and chromosome 1 of Brassicaceae. These pink blocks are meant to indicate synteny between the two genomic elements. And in Arabidopsis IncRNA, we have a long non-coding RNA originating from this region right here in between these two genes. We have a long non-coding RNA in Brassicaceae originating within this intergenic space adjacent to this protein coding gene. If we align those two link RNAs, we see very little sequence similarity. However, these two long non-coding RNAs are transcribed under similar conditions and transcribed in a similar orientation to the protein coding gene to which they're adjacent and that we're using to kind of anchor the synteny. And so this transcriptional conservation supports a hypothesis of sequence independent function. And let me describe a scenario in which you don't need sequence conservation or sequence at all to be functional. And this is, to do this, to describe the scenario, I need to describe a little bit about what we're doing with our associative techniques, where we're using linear gene co-expression networks to cluster genes with similar expression profiles across different experimental data sets. Here I'm showing you 3 different stresses, and these are time series experiments of cold, heat, and wounded leaf tissue relative to control. And here you see a module of genes with similar expression profiles that are all induced under heat. So these are most likely genes that are just constitutively turned on during heat. We have, in this module, we have 940 genes, 157 of which are long non-coding RNAs. In this bottom module, we have 250 genes that get turned on early on during wounding in leaf tissue, 2 of which are long non-coding RNAs. We would go through and we would say these link RNAs in each of these settings are responsive to heat or responsive to wounding. And then we would go through further and try to determine which of these to try and infer a little bit more contextual basis for what the link RNA functions might be. And so for these 2 link RNAs here in the wounding module, we can ask if these link RNAs are co-expressed with the genes directly adjacent to them in genomic space. So we're asking if link RNAs are clustering with mRNAs in this context. So in these experiments, these stress experiments, we got 275 long non-coding RNAs that were co-clustering with the adjacent genes, either on the 5' or 3' ends. And one of these wounding associated long non-coding RNAs is actually in a tail-to-tail orientation with a wounding related protein coding gene. And this long non-coding RNA appears to be transcriptionally conserved in Brassicaceae, but there's no sequence conservation at this between these 2 link RNAs. And we know based on, as I said, based on other work, specifically work done on this cold responsive link RNA SPALCA, that it isn't actually the sequence similarity. It isn't the sequence that's important for function here. It's actually Pol2 transcription occurring and blocking transcription of this adjacent gene that's helping to provide function for the link RNA. So these analyses provide some kind of, they allow us to make functional hypotheses that we can then go in and test in vivo. To summarize this section, I've only given you a small kind of snippets of an interesting comparative analysis that we've done for these lincRNAs in Brassicaceae. But some take-homes of our findings, we're attempting to bring parity in terms of lincRNA annotation within the plant lineage relative to the vertebrate lineage. And starting to do that, we've identified lincRNAs in 4 Brassicaceae. We've developed a short list of lincRNAs that are conserved either at the sequence level or at the transcriptional level that appear to be stress responsive, for which we can infer some type of regulatory function or capacity. And interestingly, a few that we found to be containing small open reading frames that appear to be highly conserved. And open reading frames about 30 amino acids or so that are highly conserved across the Brassicaceae or across the plant lineage, for which we would really like to understand what they're doing. During this process, we've developed some additional metrics to filter out various transcriptional artifacts to get this high confidence lincRNA annotation. And we believe that with these smaller comparative systems, we can infer function for lincRNAs based on functional characterization in close relatives. And of course, we hope to move on to functional analyses ourselves in the near future. So now I'd like to talk about, so now I've told you about a whole class of RNAs that, for which we have relatively little information. And I'd like to talk about a class of RNA modifications for which we also have relatively little information. We know that they're important, but we don't know much about what they're doing. And this, of course, are the little yellow dots or circles that I've added onto these RNAs, these example RNAs. These are the epitranscriptome that Mike mentioned. What's the epitranscriptome? Well, we've known for probably 40 years or so that tRNAs and ribosomal RNAs are highly modified. Often when you see the canonical tRNA structure, you see it just like so. But what the biology textbooks don't tell you is that they're frequently modified, chemically modified. And in fact, there's over 92 unique chemical modifications that occur on tRNAs alone. These modifications are mostly methyl groups. Here I'm showing you 2 of the most common modifications. N6-methyladenosine is this methyl group being chemically bound to this amine here on the adenosine base. Here is a methyl group being bound to cytosine to give you 3-methylcytosine. There are other types of modifications, other groups, but most of them are going to be methyl groups. These modifications occur or can occur throughout the entire transcript, but certain modifications, based on what we know right now, seem to occur within specific subregions of the transcript. For instance, M6A appears to be predominantly deposited within the 3' UTR of mRNAs, and this region-specific deposition seems to be associated with stabilizing the transcript and allowing for enhanced translation to occur. Whereas M3C appears to be deposited within the coding sequence of transcripts, and that deposition tends to be associated with degradation of the RNA. Now, I'm showing you mRNAs here, but modifications, as I mentioned up here, modifications occur on all classes of RNAs. So they don't just occur on mRNAs or tRNAs or ribosomal RNAs, but they also are deposited on lincRNAs and small RNAs, microRNAs. And these modifications occur on all bases. They're highly ubiquitous, and we know relatively little about what they're doing. Let's talk about what we do know about these modifications. And I should preface that by saying most of what we know comes from studies on M6A. Now, M6A and M3C and M5C are found on roughly 1%, each are found on roughly 1% of mRNAs in any given cell at any given time. And we know that M6A is a very ancient modification. We know that because the methyltransferase is associated with reading that mark and depositing and erasing that mark are conserved across all three domains of life. So highly conserved modification. We also know that M6A is differentially deposited in terms of tissue, stress, or even location within a transcript. I told you that M6A was predominantly deposited during or within the 3' UTR, but if you look at a specific transcript level, you start to see enrichment in other locations across the transcript. And again, where that mark is being deposited can have impacts on mRNA stability and function. As I say here, these modifications influence protein binding, they influence stability, and they influence translation efficiency. And in terms of biology, these marks we know that these marks, specifically M6A, is critical for proper development in plants. Here, I'm showing you a graphical abstract where from a few years ago, where the authors show that loss of a core M6A methyltransferase leads to improper shoot apical meristem expansion. And this is not kind of a result of systemic loss of M6A marks, but it's actually the result of loss of a few marks on a few specific transcripts that leads to this improper expansion. So M6A is incredibly important. We know this not only in Arabidopsis, but we also know this from work done in humans and mice. But M6A is just one of over 100 modifications, of which only a few have been examined really in all eukaryotes. So we know relatively little about the epitranscriptome. In particular, we know little about when and where these marks are applied and the impact that they're having on the transcripts that they modify. And in terms of a evolutionary context for these modifications, we know little about how these marks are conserved and whether the sequences, the motifs that they are applied to are conserved across the plant lineage. And again, we don't know much about whether modifications are conserved between orthologs across a lineage, or if these modifications influence gene evolution. And if these modifications impact RNAs in similar ways across species. Really, we have very little information on it from a comparative perspective. To address this gap in our knowledge, the lab, or my lab, has really two projects ongoing. One is a collaborative project with Dr. Brian Gregory from the University of Pennsylvania, Dr. Eric Lyons from University of Arizona, and Dr. Rebecca Murphy from St. Mary College of Louisiana. And this project is taking a high throughput approach, utilizing publicly available RNA sequencing data to identify and characterize the epitranscriptome across the plant lineage. This is the source of a Tiara Tech that was recently awarded. And then the second project is taking a much narrower view and a much more fine scale view of looking at modifications within the Brassicaceae and how those modifications are influencing plant stress responses. I'm going to talk to you very briefly about about project one and how we're going about identifying modifications, the epitranscriptome, utilizing publicly available RNA sequencing data. So this is, I think, maybe the second time I've shown this this table here. Maybe it's the first. But you have, many of you, if you've heard me talk, you know that I like to utilize publicly available data because it saves money, and there's a lot of information that's stored in those data because each of us within the community is perform those RNA sequencing experiments for a specific reason. Many of those reasons are an attempt to look at how plants are responding to stress. So there's a lot of useful information out there, and we can examine all of this RNA sequencing data, roughly 300 terabases of RNA sequencing data, using this computational algorithm HAMMER that Brian Gregory's lab developed about 7 years ago. And HAMMER is capable of identifying 45 unique RNA modifications from RNA sequencing data. And it does this taking advantage of the way in which, I guess the pattern in which reverse transcriptases interpret modified bases. So if a reverse transcriptase that you're using in your RNA-seq or cDNA library preparation comes across a modified base that is that's modified along the Watson-Crick base-pairing edge, it's going to see that modification and it's going to think that this specific modification is actually, or this base is actually a different base than what it is, and it's going to incorporate in the complementary position the wrong nucleotide. When you look at this RNA sequencing data, when you align this RNA sequencing data, this appears as a mismatch or a SNP in your RNA sequencing data. And it's the frequency or the pattern of mismatches that Hammer is utilizing after it screens for quality score and biallelic frequencies. It's this pattern of mismatches that Hammer is using to infer a modification. And just a real life example of what one of these inferred modifications looks like is if you have, so we have a uracil here at this position. We have a lot of RNA sequencing reads stacked up at the site, and we have kind of a pattern of G's, A's, and C's, this frequency of which Hammer utilizes to infer that this site is actually a modified uracil where a methyl group has been bound to the third position. And of course, all of this is based off of training that Hammer has, training of tRNAs and other RNAs that we know are heavily modified. Again, we do all this identification, but we're not just identifying for the sake of identifying. We'd love to know what these modifications are doing. So we're going through and performing very similar, asking very similar questions to what we were asking for the link RNAs. We're asking questions about context-dependent deposition. So are each of these different marks deposited in different regions of the mRNAs or of any of the link RNAs we're looking at? Again, each of these modifications we are looking at, each of these 45 modifications, we have very little knowledge or understanding of where the marks are deposited and what they're doing. So we would really like to know where they're being deposited and if these modifications are getting applied during a specific developmental time point or a specific stress. We also would like to know if we can infer biological roles for these modifications based on the context under which they're applied. Do we see an increase in a modification under a specific stress experiment? And do we see the frequency or the levels of that corresponding RNA go up or down in those same contexts? And then we would also like to know if these modifications are conserved. We want to know if these modifications occur on the same transcripts or conditions across species, so that we can generate this this rule book for what these modifications are doing across plants. Now, the one caveat or the one drawback of Hammer is that it can't identify modifications that fall outside of the Watson-Crick base-pairing edge. And unfortunately, M6A and M5C, two of well, what we know are the two of the more common modifications, are outside of that Watson-Crick base-pairing edge. So they don't actually cause errors during reverse transcription. So to identify these marks, typically what we have to do is we have to do RNA IPs using an antibody for M6A, or we have to do bisulfite sequencing of mRNAs to get at M5C. These are incredibly tedious experiments, and they typically predict. They usually only recover about 20% of modifications. So to ask some more fine-scale questions about the roles of M6A and M5C in a comparative framework, we're looking at these two modifications within Brassicaceae. And I'm just going to reintroduce 4 species I've already talked about: Arabidopsis, Camelina, Brassica, and Eutrima, all close relatives or all relatives within the Brassicaceae. And Camelina and Eutrima just happen to be two heat and salt tolerant representatives within the family. And then in addition, Camelina and Brassica have undergone recent or relatively recent whole genome duplication events. So now we have a comparative framework under which we can ask a few different questions that we can't quite ask under such controlled conditions in our HAMMER project. So the first question is of course, do species with differential stress tolerances display unique patterns of RNA modifications? And are the RNA modifications differentially deposited among paralogs following whole genome duplication? Do stress responsive genes, does a stress responsive gene in Arabidopsis that's for which we have orthologs in Brassica, do we see differential modification and potentially differential function for those genes in Brassica or Camelina? And so this framework is allowing us to compare really between species, between different genomic events, and between different stresses. And we're getting around some of the technical difficulties with RNA immunoprecipitations or bisulfite sequencing by performing Nanopore direct RNA sequencing. Now, performing direct RNA sequencing means that we don't actually convert the RNA into cDNA. So we're retaining all of the information that's found on that RNA molecule, i.e. all the modifications that are found on that RNA molecule. And we're taking advantage of, again, essentially of base calling errors from the Nanopore base calling algorithm that can't quite interpret the little squiggles, the little signal intensities that change when it encounters a modified nucleotide. So just to remind you of what is kind of happening, a very simplistic cartoon of what's happening during Nanopore sequencing. Of course, we have these proteins that are helping to funnel an RNA through a nanopore. And it's really this current that's coming across this membrane that changes each time a different base is read through. As you see here, it's this current that's being read and then being base called. And again, it's changes in the currents based on the modification that's telling us that something is going on at this specific site. Now, we can't specifically say whether or not it's M6A or M5C unless we have a tissue or we have a genetic background that is lacking or knocked down for one of those modifications. So we're using an Arabidopsis methyltransferase mutant, MTA mutants that's deficient for M6A to train this epinano or Tombo model. And then we're hoping to apply this trained model to the other three species that we're looking at because we don't have mutants for any of the methyltransferases in Camelina, Brassica, or Eutrima. Now, we've performed the kind of systemic heat and salt stress experiments, and we've performed the Nanopore direct RNA sequencing in each of these species or genetic backgrounds. And we are currently in the process of analyzing these RNA sequencing data for modifications. So hopefully I'll have more to talk to you about soon. Okay, so I've told you now about novel transcripts for which we don't really know much about. And I've told you about the epitranscriptome, which we know is important, but we don't know completely what each of the different marks are doing. And lastly, I'd like to bring us back to something we're all very familiar with and we're all comfortable with, and that's utilizing changes in RNA abundance to get at or to understand mechanisms of stress resilience. And this work is really primarily a collaboration with my cluster partner, Dr. Magda Wielkowska, who's I would say, a very excellent plant physiologist and phenotyping expert. And she's going to be talking in a couple of weeks. So I'm not going to talk about really any of the phenotyping or the physiology, but what I would like to talk about are the different ways in which we can utilize RNA-seq to get at molecular mechanisms. Of course, RNA-seq, many of us have used RNA sequencing in different levels of experimental complexity to get at our kind of candidate genes in which that we then go in and target for genetic assays or et cetera. And so, of course, the most simplest, I would say, of experiments that can be performed with RNA sequencing are, of course, the pairwise comparisons between a couple of genotypes or a couple of stresses. And then, of course, we can get a little bit more complex and we can compare between multiple stresses or genotypes and really perform something like hierarchical clustering to get at specific subsets of genes that are enriched in a specific background that we then perform, go enrichment or something to make functional predictions. And then, as I mentioned earlier, we can get even more complex and we can do things like co-expression networks to get at, again, these modules of genes that are enriched under specific conditions. And then, of course, there's what I would consider perhaps to be the most experimentally complex, which would be associating genome-wide association studies with transcription or transcriptome-wide analyses to identify genes that are significantly associated with specific phenotypes. And just to give a preview, we're looking at phenotypes in, obviously the Brassicaceae, as well as the Solanaceae, looking at wild relatives of some of our most favorite model systems. But yeah, how are we going to go about, I guess, how are we going to go about getting to phenotype? What are the phenotypes that we're monitoring and how are we doing this? And again, I'm not going to go too much into phenotyping because that's not my specialty, but my second hobby after RNA biology is computers. And what I'm showing you here is an extremely small computer. It's a Raspberry Pi Zero. I don't know if any of you still remember the days of the iPhone 4. Many of you maybe are looking at the iPhone 12 right now and salivating over it, the idea of getting one. But this Raspberry Pi Zero has similar specs to an iPhone 4. So it's a very small. This is sitting on a post-it note about 3 inches by 3 inches. So this is about 1.5 inches wide. And it's significantly cheaper than the Bankbuster iPhone 4 at about $10 per Raspberry Pi Zero. And we're using these Pis to control cameras on these little flat phenotyping rigs where we are taking pictures. We have 2 cameras per rig. And we're taking pictures every 30 minutes to monitor changes in development of these plants. Here you see them in the growth chamber. Again, right now we have 9 of these set up. We envision setting up many of these rigs, and they allow us to monitor plant growth over time. And just showing you here kind of changes in the plants as they're developing. These are pictures taken every 30 minutes. And what you get from this is, or one of the most simple things that you can get from this is kind of change in pixel density over time as the plants are growing. And you can compare this to plants that have been stressed or exposed to a specific stress. And you'll see that basically we can use this high temporal resolution phenotyping to identify regions that are important, or developmental time points that are important for molecular analyses. So this is helping us to pinpoint when we should be taking samples for RNA sequencing and then comparing it back to the phenotypes that we observe, that Magda is observing. I'll just wrap up to in conclusion, say that I hope that I've exposed you to a few new aspects of RNA biology today and helped you to appreciate the complex ways in which RNAs can control mechanisms of stress responsiveness, both influencing what's happening at the DNA level, influencing what's happening at the translational level, and also really what's happening at the RNA level. So with that, I should stop and thank a few people. Several people associated, including Magda, associated with what I call the Polska Bandit, the Polish bandits. Where we've branded ourselves the Stress Architecture and RNA Biology Lab. Some important people that were associated with either the linked RNA story or the epitranscriptomic story are listed here. The new third person in the cluster, Ola Skiric, working with her and with Magda on an RNA compartmentalization project looking at stress granules. And of course, I should thank you for your time and the NSF for funding. Thank you. And if there are any questions, or if you have any questions about my interests in adjunctship with plant bio or with the field, please fire away.
So just unmute yourself if you'd like to ask Andrew a question, or you can throw it into the chat.
Andrew. Oh, go ahead.
Go ahead, Wojtek.
So I don't like the term banda, but my question is...
That's Magda's doing.
I suppose so, yes. But my question is, when you this concerns the non-coding RNA parts, what do you think is the role of the secondary structure of those link RNAs, if any? So like you mentioned that in some cases there's just the transcription complex assembly, that's what it's needed. But do you think that there is this, I mean, some of them has been shown that they have secondary structure that's important. I wonder how prevalent this is, you think?
I would argue that, I mean, I hate making blanket statements about a whole class of RNAs, but I would say that structure is definitely important for let's say a significant number, because obviously you need there. So we've shown with Brian Gregory and obviously many others have shown that RNA structure is important for the binding of RNA, some of these RNA binding proteins, HNRMPs, et cetera. And structure we think is important for potentially important for recruitment into stress granules and yeah. So I think maybe you're getting at the degree to which we expect structure to be conserved. Is that
that would actually be part of it, yeah.
Yeah. So we don't have a lot of information about structural conservation outside of a few examples in plants, but we do know that there's definitely some selective pressure on structure being retained in vertebrate systems where they have these massive genome-wide alignments, and they can actually look get a good sense of the evolutionary pressures there. So I do think that structure is important for some of these linked RNAs, but I don't have a good idea of how many exactly.
Thank you.
So Andrew, I thought that was a great talk, and thanks very much. I have a question. So some of the covalent modifications of the RNAs you talked about, and some of the structures, and potentially the small ORFs, do they, have you noticed any association with the potential for trafficking and flow mobility?
Ooh, this is something I've talked to briefly about with Margaret. So I know that a few of the linked RNAs that we've identified show up in a data set produced several years ago now that was looking at mobile RNAs. I don't know how, I think this was mobile RNAs in Arabidopsis. And I don't know how, I would need to look at that to see how reproducible that was. But I do know that based on again, mammalian literature, that modifications can be important for trafficking, especially of the smaller, the microRNAs. So I think that would probably be where we would get better traction in terms of experimental evidence. I think it's relatively easier, or it's easier to look at with Margaret's grafting experiments, for instance, to look at trafficking of microRNAs than it would be, and then to get a sense of the modifications that are occurring there. So that is definitely something. I think that those modifications are important for trafficking within the cell, and I wouldn't be surprised if they were important for trafficking between cells.
Okay, thanks.
There's a question from Adrian in the chat. Nanopore sequencing is known for having non-random sequencing errors. Do you think a high proportion of these errors are really these types of modifications?
That is an excellent question. That is something that the authors, so there have been several recent publications on inferring modifications from Nanopore RNA sequencing data. And of course, Nanopore would love you to think that all of those SNPs are a result of modifications. I don't think that's the case. Even though they have improved their sequencing technology quite a bit, there's still some random errors. But I think the take home from some of the more recent publications say, if the error rate is like 3 or 5% or something right now, I think a significant portion of those can be attributed to non-random, like specific within the RNA sequencing. It's specific errors that may be results of modifications, not just of M6A and M5C, but some of the other many other modifications. So the group that was behind M6A, there was a recent Arabidopsis paper, and there's I think a 2019 paper in mammalian systems, and there were a surprisingly high number of M6A marks that they identified through Nanopore RNA sequencing data, and they demonstrated that these marks were specific. They were M6A because they had the mutants to compare against. So I think Nanopore is going to be a really strong resource for us.
Another question from Jeff Doyle in the chat. You mentioned polyploidy in Brassicaceae. Could you elaborate on specific questions you want to address about polyploidy using your system?
Yeah, so I think one of the interesting, I have no strong rationale for why I'm asking this question, but I think it would be interesting if after whole genome duplication events where you suddenly have two genes. Back up. I think it would be interesting if the epitranscriptome was somehow involved in sub or neo functionalization or pseudogenization, particularly of stress responsive genes. Again, I have no. I think Brian has seen some interesting stuff in wheats where you have kind of whole subgenomes experiencing preferential modifications, but I think it would be a fascinating aspect of the ways in which genes evolve. So.
Great, thanks.
Other questions? Anyone?
I'm curious about using Hammer to identify the PTMs and whether when you're using data sets from multiple sources, there's a higher probability of misidentifying SNPs as modifications.
Yeah, so some of the technical details. So we need fairly deep RNA sequencing. So I think we need within the transcriptome we need 20x or so. So we're only really confidently calling in the genes that are typically most highly expressed. In addition, we're not combining multiple experiments into the same Hammer analysis per se. We're comparing between analyses. So let's say we have 10 leaf control. Everybody does Arabidopsis or uses Arabidopsis seedlings for their experiments. So we have a lot of RNA sequencing associated with that. Do we see across a significant proportion of these experiments the same genes getting modified in the same tissues under the same conditions? And then we compare that between tissues or conditions. So
cool, thanks.
Yeah.
Any other questions? Okay, let's thank Andrew one more time. Thanks a lot, Andrew. Great talk.
Thank you.
This has been a production of Cornell University, on the web at cornell.edu.